Good morning, good afternoon, good evening, and welcome everyone. I am Carmen Guguta, Head of Business Development and Marketing at Technobis Crystallization Systems. Today, we are happy to bring to you the webinar on influence of polymers on nucleation and growth kinetics of pharmaceuticals, a webinar presented by Dr. Kautar Abu Sharif from Eli Lilly. For one reason or another, I always have a problem with the first, with going to the second slide. I'm sorry for that. This webinar is being recorded and I will make it available to you tomorrow. You will receive an email from us with a link to the recording. The recording will be available on our website all the time, so you will be able to freely watch it whenever you have time afterwards. You are being muted, but if you have a question, you have something that you want to share with us, please make sure you can use the chat function of this platform. Or after the webinar, write us an email at info@crystallizationsystems.com. If you have a question for us or for our presenter, we will make sure that our speaker today will answer all your questions. The webinar will last about 45 minutes, I think, including questions. It is great to see many of our customers, current customers, joining us today who are users of our crystallization technology and are quite familiar with Technobis crystallization systems. For those of you joining us today for the first time, Technobis Crystallization Systems is a Dutch company based in the Netherlands, in the north of the Netherlands, focused on developing innovative instruments for applications, including solubility screening, crystallization process development, and formulation. I'm proud to say that we have more than 500 systems installed in the field all over the world, from USA, Canada, to Europe, to Asia, India, China, Japan, and even down to Australia and New Zealand. Our instruments, as I mentioned to you, are instruments for crystallization, process development, and formulation. Featured in during our webinar today are both our Crystal 16 and crystalline instruments. Those are multiple reactor systems that combine heating, cooling, and steering and transmission technology within each reactor. The Crystal 16, the one that you have here on the left side, reactor system with 16 reactors, specifically designed to work at 1 milliliter scale. You can easily generate temperature-dependent solubility curves with a little bit of material into up to 4 solvents in one run within 4 hours. You may perform any type of solvent screening, polymorph, salt formation, co-crystallization type of screening. You can construct phase diagrams, any type of solubility metastable zone determination. Crystal 16 is your instrument. Once prepared with your solid-state roadmap created from the Crystal 16, the crystalline instrument, what you have here on your right side, will help you to develop and optimize your crystallization and formulation process with the additional analytical capabilities such as the particle viewer, the camera, will allow you to actually see what is happening in the vial while you are performing a reaction or an experiment. You can see when crystallization occurs, determine particle size distribution, assess crystal habit, evaluate the stability of your formulation, you name it. If you have a Raman spectrometer in your lab, you can easily connect it to this crystalline instrument, and you can follow the same way you would follow, for example, with a Raman spectrometer, chemical transformations, reactions, or polymorphic transformations. The crystalline has 8 reactors and works at 5 milliliter scale. It is the instrument that will provide you the information you need in order to optimize and troubleshoot your crystallization or formulation process. If at the end of this webinar you think that you would like to have or to try one of these instruments in your lab, please, we will be happy to work with you and answer any questions that you may have related to the instruments or any type of capabilities of those instruments related to your specific application. We will be happy to answer your questions. You can either write to us, as I mentioned in the beginning, at info@crystallizationsystems.com, or quite easily via the website, you can get directly in contact with us. We would be happy to help you. And now I'm very happy to introduce you our speaker today, Dr. Kautar Abu Sharif. She is a senior consultant engineer at Eli Lilly, and she also is the group leader. She leads the group of bioproduct research and development at Lilly Innovation Center in Cambridge, Massachusetts. Dr. Abu Sharif received her PhD in 2014 at the Purdue School of Chemical Engineering under the Professor Jim Lister, and having Professor Lynn Taylor as co-advisor, co-supervisor at the School of Industrial and Physical Pharmacy. Coco, thank you very much for joining us today and sharing your knowledge and expertise with us and with our customers and our attendees today. I think you should be able now, just 1 second, to share your screen.
Thank you, Carmen. Are you able to hear me clearly?
Yes.
Excellent. And let me know when you can see my presentation in full presentation mode.
I see your slides. Now is in full presentation mode. Now you're good to go.
Thank you, Carmen, for the kind introduction, and thank you everyone for attending this webinar. As Carmen mentioned, what I'll be talking about today is the influence of polymers on nucleation and growth kinetics of acetaminophen, or APAP. This work was done a while back, actually, during my PhD at Purdue University. We do have a publication that you can refer to, as well as my thesis, for a lot more detail than what I'll be sharing with you today. So the agenda of the talk is as follows. I'll go over the objectives of this specific talk. I'll discuss materials and the methods, and then the theory behind the work. And I'll end with results and conclusions. So the purpose of this work was twofold. Number one was to try to explore methods that can help us evaluate dissolution, nucleation, and growth kinetics of APAP as a model pharmaceutical compound using instrumented small-scale crystallization vessels in the presence of various polymers. The second goal was to determine if there are any correlations between the crystallization and inhibition effects of these polymers in both the solid state as well as in aqueous supersaturated solutions. And the reason why is because, you know, the drug normally would end up being in a solid state in a tablet or a capsule. However, when it is administered orally, it will turn into a solution state. So you really want to understand both the impact of these polymers in both states. And we could reach supersaturated conditions very easily in the GI tract, especially for poorly soluble compounds, right? If you have a weak base, for example, going from the stomach, where you have a very acidic environment, into the duodenum, the drug can experience major pH swings that could lead it to precipitate out, whether as crystalline or amorphous. One thing that I want to point out here is APAP is just the model compound used for this study. You can find more information on how we've used these methods with dipyridamole, and I left the reference below. So I'm going to talk now about the materials and methods used. As I mentioned, APAP is our model compound, and here I show the various polymers that were of interest to my co-advisor. So we really wanted to see, are these polymers going to inhibit or speed the rate of both nucleation and growth. And I'll talk about how we try to deconvolute the two in the next slides. Now, what you see on the right portion of the slide are the two methods that we used. We used the crystal 16 parallel crystallizer. This is a setup where you can actually put in 16 vials, as the name says, and you could stir your vials, you could heat them, cool them, and there is light transmitted through the vial. So the way how we measure these induction times is by tracking the transmissivity changes across each vial. And I'll talk about this in more detail in the next slide. The second instrument that we used is the crystalline PV, and we used it in order to measure or count the number of crystals as precipitation is taking place. The crystalline PV really offers a lot more insight than the crystal 16, because you're actually able to view what's happening inside your crystallizer. So I want to discuss a little bit of the theory of how did we get these nucleation rates as well as the growth times from induction times. And I want to walk you through this process step by step, so that if you are an owner of one of these instruments, you can apply the theory directly and get some insight as to what your nucleation kinetics are in whatever system you are studying. So you can add whatever polymer of interest you have, or just the drug or the substance of interest. So the theory here was really inspired and adopted from the work of Professor Joop Horst, and I did leave the reference here for you to go and look at the paper in more detail. But basically, we all know that nucleation is a stochastic process. So we assume that the probability of forming a nucleus is going to follow a Poisson distribution, where here P is the probability of forming a given nucleus. PM is the M refers to the number of nuclei, and that equals the average number of particles forming within a certain time interval divided by M, and then the exponent of minus N. So if we look at the probability that there is at least one nucleus, that equation becomes as follows. So then what we're really interested in is, what does this N mean and how is it related to your nucleation rate? So we always go by J, the definition of J, but if you flip the equation, the number of nuclei that you have in a given solution volume is J, which is your nucleation rate, times the solution volume times induction time. And what is induction time, or this nucleation time? The induction time here, we defined it as the time interval between the birth of the first nucleus and the growth time. The reason why we defined it this way, or it was defined in the paper this way, is because you cannot see when the very first nucleus forms. So you have to allow enough time for the nucleus to grow in order for you to detect it, and in order for the solution to make enough of an impact on the transmissivity of the light for you able to detect any changes in that light transmissivity, which is really what you're tracking at the end of the day with the crystal 16. So in order for us to get the nucleation rate as well as the growth times, what we did first is we captured the induction time from the crystal 16. We built this probability distribution function, and from that we were able to estimate J and TG using simple parameter estimation. So the way how we conducted these induction time experiments is as follows. First, you have to prepare your solution. We had different polymers, as I mentioned. So first we tried to make a supersaturated solution of APAP. We prepared 20 mils in pH 6.8 sodium phosphate buffer. our supersaturation ratio was 1.5. This is the initial supersaturation ratio. As precipitation takes place, that ratio is going to change, obviously. But we took that initial solution. We started at 700 RPM, heated at 70 degrees. We added the polymers, and then not all of them at once, separately, as we are interested in how each one impacts nucleation and growth. Filtered the solution, and then we put aliquoted it in various vials. These are like really HPLC vials, small volume, so you don't need such a large batch. Then we stirred the solutions in the vials at 700 RPM, heated them to 60 degrees at 20 C per minute. The reason why we did that is because we really wanted to ensure that our solution is, when it's heated, everything should be dissolved. So we held it at 60 degrees for 4 minutes, like I said, to ensure complete dissolution. And then that's what defines the clear point. And I'll mention that, or I'll go over that in the next slide. It's really important to know when is your clear point and when is your cloud point, because that is the actual definition of induction time. Then we cooled down the solution to 20 C at 10 degrees C per minute. And we repeated the cycle pretty much a couple of times. So you are forming precipitation, or you're forming these crystals pretty much just by cooling crystallization. The reason why we repeated these is because for us to build that probability distribution function, you really need a lot of time points. So we did 80 experiments per polymer in this system. So as I mentioned earlier, the key parameter that we need in order for us to build our probability distribution function is the induction time. What I'm showing you here is just an example of the transmissivity curve. This was actually taken from another experiment, because in this case, we actually heated our compound up to 60 degrees. This was a different experiment where I heated the sample to 37. We kept it at 37 for a couple of minutes, and then as the system was being cooled down, you can start seeing that the drug or the transmissivity is dropping. So this is how we define your clear point, and this is the cloud point. The left is temperature. This is transmissivity, which is really an optical property of your material, and here is the time. So the nucleation induction time in this scenario is really the time elapsed between the onset of supersaturation and the detection of the new nuclei. So we calculated it as the difference between that initial decrease in light transmissivity, and this is only happening because your solution is now becoming cloudy, and that's due to nuclei forming and growing enough to make a difference in the light transmissivity. So as I mentioned, we repeated the experiments for about 80 times per condition or per polymer. And what you see here is a true reflection of that stochastic nature of nucleation. You repeat the same experiment under the same conditions, and every time you're going to get a different induction time. So based on these data, we formed those probability functions that I mentioned earlier. What I'm showing you here is the probability function as a function of time in the presence and in the absence of the polymer. The blue marker here is just APAP. I want to remind everyone the initial supersaturation that we chose was 1.5, and that was kept the same across all polymers. And then I'm showing the probability for PAA, HPMCAS, and then HPMC. So what you see here actually is that PAA had a very negligible impact on the induction time or the nucleation kinetics of APAP. HPMCAS and HPMC had an intermediate effect. What you don't see in this graph, or what's missing, is PVP and PVPA. And those polymers actually retarded nucleation for so long that even up to 2 days, there were no nuclei forming. So that's just, you know, the primary data that you can get out just by looking at how these induction times varied across the different polymers. Now, what I mentioned earlier, our key objective was to actually back calculate the nucleation rate at that particular starting supersaturation level. So what we did is I built a, I coded the equations in GPROM's model builder, and I created a user interface because I was doing this for so long that it just made sense to automate the process. What I'm showing you here is just a very basic user interface where you put the number of experimental data points. Let's say in this case you have 80. You put all of your induction times in whatever order, the number of bins, and then you can put in an initial estimate of your primary nucleation rate, the growth time, the volume of the solution, and some lower and upper bounds of your induction times. And basically you run a parameter estimation to get the values of the nucleation rate as well as your growth time. Those values are, once the model converges, what I'm showing you here is the simulated probability distribution function using the final outputs of your nucleation rate and your growth time. So we see that there's really good agreement between my simulated as well as experimental values. And like I mentioned earlier, you could just look at the curve and that would tell you some information about how the different polymers behaved and how did they impact the nucleation kinetics without really getting these values. So if we look at the table, the key thing that I want to point out is that, like I said, PVPA and PVP were the most effective at inhibiting nucleation altogether up to 2 days. But what we see here is PAA, HPMCAS, and HPMC. So the polymer that had no impact is PAA, and you can see that here. You can compare the nucleation rate of it versus in the absence of any polymer, right? So we see that it actually made it worse. It actually promoted nucleation. So that's really one of the conclusions is PAA promoted the APAP nucleation. As I said, PVP and PVPA were the most effective, but HPMCAS and HPMC had this intermediate inhibition effect. They weren't the best, but they didn't promote nucleation. We really think that this nucleation inhibition effect is likely due to the intermolecular interactions between the solute molecule and the polymer, but also that these drug polymer interactions can compete with the solvent drug interactions, and these are likely very important for us to understand what is happening in this complex matrix, because you don't have just the polymer with the APAP or the drug by itself. It's a tertiary, or it could be a more complex system than that, where you have to worry also about the buffer composition, etc. So one thing that I want to point out is that those nucleation rates were obtained at the initial supersaturation that we started with. So that J value is calculated at a very specific supersaturation level. What we were interested in is also monitoring how the nucleation rate varies as the system is precipitating out. And we wanted to get an idea of as precipitation is happening, are we seeing the crystals grow? Are they agglomerating? What's happening inside the crystallizer? Induction time is important, and knowing what polymers retard or promote nucleation is important, but it's good to get even better insight as to what's happening as the system is precipitating. So this is where the crystalline PV came in handy. I ran 4 milliliter solutions that were prepared exactly the same way as I mentioned in the earlier slide. All of the experiments were done in triplicate, and the crystal images here were captured every second during the course of precipitation. Now the key is what to do with those images or the videos once they are obtained. So this is where I analyzed the images just in ImageJ using the Yen algorithm to extract the particles from the background. In the next slide, I'll show you a video of what this really looks like, and also what does the original image look like versus the image after thresholding. These thresholded images were analyzed to count for the number of crystals per 2D image as a function of time. As I mentioned earlier, what we are really interested in is the nucleation kinetics. How are they changing in the presence of polymer? So the assumption here is that the change in the number of counts as a function of time is directly proportional to your nucleation rate. So every image you start seeing more and more and more crystals showing up. That means that is your, if you count the number of crystals per image, that's a, can be directly proportional to your nucleation rate in the entire solution volume, because this is a mixed system, and hopefully what passes through the camera is representative of what's happening in the rest of that vial. So what I'm showing you here is the crystal count number per 2D image as a function of time of just APAP in the absence of any polymers. The one thing that I want to point out is, as I mentioned earlier with the induction times, nucleation is a stochastic process. So every time you run the same experiment, same conditions, you will get a different induction time. And you see here in this first run, the system started to crystallize after 200 seconds. Here's like 400, here's 800. But also, so when you look at the slope of this line, this is showing you how the number of crystals is increasing per 2D image. So that's really a reflection of nucleation taking place, more and more nuclei forming. What is also interesting here, and what I did not expect, is this drop. So initially, if you are not looking at the images, you may think that the system is dissolving, or that something is happening. Either the nuclei are precipitating and just are stagnant now at the bottom of the vial, and what is passing through the camera is, is becoming more clear as time goes on, right? It almost seems like stuff is dissolving or sticking somewhere or absorbing into the vial surface, and the solution is becoming more clear. It's hard to tell what is happening without also measuring the concentration of the solution as this is taking place, but looking at the images is gonna offer you a lot more insight, as I said. What we ended up seeing is that, unfortunately, here the system was actually agglomerating. When you have enough nuclei formed, they start agglomerating, and what used to be 5 particles are now just 1 particle because they're all sticking together. Some polymers, I'll show you in the next slide, across the various polymers, we saw some where the system does not agglomerate. So what I'm going to show you here is an actual video of the solution and how the experiment happens. So this is in the very beginning where we have crystals in solution. As I mentioned, we always take the solution, we heat it up to 60 degrees. So here you start seeing, and please keep in mind that this is like sped up. The system here is dissolving. What you see are just some of the either dust or some particles, not APAP, but other things that were in the vial, just some impurities in the system. And these are really insignificant. It's like 1 per 2D image. When a real nucleation happens, you will see many, many, many crystals, and it's clear that nucleation is taking place. So now the system, oh here, there we go. You see how the system is now nucleating. You have many, many crystals forming. So one key to point out is there are a couple of things happening here. The system is being stirred, but also we are undergoing this heating cycle. We keep the solution at the maximum temperature to make sure that everything is dissolved. And that's when we define our clear point. And then we cool the solution, and that's when you start seeing these crystals form. As I said, we had to repeat this a couple of times. So this is why you really want to ensure that everything dissolves completely. The very first experiment started with an entirely dissolved solution with no crystals. But once you pass that heating and cooling cycle, you have to keep repeating it many, many, many times. Okay. So here I'm just going to show you some of the results from this nucleation experiment and how we utilized image analysis. One thing I failed to say here actually is this is the original image as captured with the crystalline PV camera, and this is the thresholded image. And you see that all of these shadows, etc., are actually not being captured. This is often a question, is how do you make sure that you're not really capturing anything that's in the background? And the algorithm I made sure was put together so it doesn't take into account, or it filters out all of the background. So what I'm showing you here are the results across the different polymers. First one, graph A, is in the absence of any polymer. This is just APAP and the induction times as well as the nucleation rates that we obtained. B is HPMCAS. And what you see here, like I mentioned earlier, there was no agglomeration actually. You just see nucleation taking place and we're sort of steady, or we reach a plateau. Now for C, that was PAA, and we see something very similar to what we saw in the absence of any polymer. D is in the presence of PVP, and E is in the presence of PVPVA, and F is the HPMC. So by just looking at this curve, you can tell by comparing the time which polymer was good at inhibiting nucleation and which polymer was not, in comparison to APAP by itself. So here are the crystal count times and detection times summarized for you from those graphs in this table. What we will be comparing first is APAP versus APAP in the presence of PAA. So what we see here is that the slope of that line, or the slope of the line of the crystal counts per 2D image, is very similar for APAP versus PAA. This is the average slope across the three replicates that we did. Now looking at the detection times, they're somewhat similar as well. So fairly negligible impact if we compare it to, say, PVP. So PVP here was the strongest polymer at slowing the nucleation rate. And you can also look at the induction times. Here we're between 150 to 785 seconds versus for PVP, we're between 570 to even up to 8,000 seconds. PVPVA, while it retards the onset of nucleation, it's actually not the best at slowing the rate of nucleation in comparison to your cellulosic polymers. And that was really interesting to see here. So if you look, you see that the nucleation rate is lower. However, once the crystals are forming, they form really fast. Now, something else that we were interested in, I covered all of the nucleation kinetics. We were also interested in looking at the impact of polymers on crystal growth. So what we did here is that we assumed that the crystal growth of the added seeds is going to be largely responsible for the desupersaturation that we see in the various systems. So this is a seeded crystallization experiment, and we try to operate within the metastable zone width so that we can ensure that any decrease in supersaturation after adding those seeds is not due to nucleation and is mostly going to be attributed to growth of those seeds that we added. What I'm showing you here is APAP in the absence of any polymer. Those are your orange circles, and then APAP in the presence of the various polymers that we've been studying. This graph just shows the concentration as a function of time, and it shows you how the system is desupersaturating and how that rate varies as a function of the different polymeric systems that we are using. We see that PAA again had very little effect on APAP crystal growth, very similar to the drug by itself. Other polymers were more effective at inhibiting the crystal growth, especially HPMC as well as PVP. This apparent effect of the polymers on the growth rate is much lower, we think, than their effect on inhibiting nucleation. I wish I could show you how the desupersaturation curves look like in the crystalline PV or the crystal 16, where we just had nucleation impacting the desupersaturation. I don't have those measurements, however. So lastly, what I'm going to talk about is the impact of polymers on crystal morphology. This was really interesting, and my colleague Niraj actually did this part of the study where we looked at micrographs of APAP crystallized in these different polymeric systems, and what you see here is pure APAP shown in this image A kind of produces thin, flat, elongated crystals, and these change dramatically in the presence of different polymers. So PVP in figure D didn't change the morphology but much, but the crystals were a bit smaller, well, not a bit, actually distinctly smaller. In figure C, what I'm showing you is APAP in the presence of the PAA, and these crystals were very different looking than in the absence of the polymer, where they were larger, prismatic, and polyhedral in shape. Figure B is showing you the impact of HPMCAS on the crystal morphology of APAP, where now the crystals are kind of like rod-shaped in a sense, slightly different than in the absence of polymer. And then in figure E, what we're showing you is the crystals in the presence of PVPVA, where the crystals were large and they had this parallelepiped shape. And then figure F is the last one, where in the presence of HPMC, where we saw the greatest inhibitory effect of the polymer on crystal growth. The crystals were very small and they had a poorly defined morphology in comparison to APAP by itself. You can see here the comparison. So we know that polymers are adsorbed preferentially into specific phases of the crystal and depending on the direction of the strongest hydrogen bonds. The attachment of the polymer to the fastest growing phase can actually result in a decrease of the measured growth rate of your compound in the presence of that particular polymer. So I hope that at this point I showed you how you can use the crystalline PV and the crystal 16 along with microscopy and simple desupersaturation experiments to really get an insight on how your model compounds behave in the presence and absence of different polymers. These methods can be applicable to any system. What I noticed from this study is that the crystalline PV in comparison to the crystal 16 offered much better insight on secondary nucleation kinetics, where in this instance PV-PVA may be good at inhibiting primary nucleation, but wasn't as effective at preventing or decreasing the rate of secondary nucleation of APAP. Considering the impact on crystal growth, this is kind of like the rank ordering in the solid state. Sorry, not in the solid state, in solution. One of the objectives that I said is we wanted to compare how these polymers also behave in the solid state. So when you look at intercooled melts, the rank ordering of these polymers was actually different, where PAA was much better than PVP, and so on. Kind of like the opposite of what we saw in solution. And we think that's because PAA is ineffective in solution because of its ionization at pH 6.8, and it cannot form hydrogen bonds, which are important in inhibiting the growth rate in the solid state. The other thing that I want to point out is that this inhibitory effect of these polymers for the amorphous drug in the solid state can be different from its effect in solution. It's worth studying both. And also, if you want to inhibit crystallization from happening, it could be worth it to consider a combination of these polymers, not just a single polymer. With that, I would like to thank my previous advisors, Dr. Litster and Dr. Taylor, as well as my collaborator, Dr. Tracy. All of this work was done at Purdue University, and thank you for attending the talk.
Thank you very much, Coco. I really appreciate that you have joined us today and you've shared your knowledge, your experience in this nice case study with us. It was also quite nice to see the picture of the crystal 16, and I haven't mentioned this to you, or even today I didn't mention it. That was the first version of the crystal 16. In the meantime, have also a second version, and this year we are celebrating actually 16 years of crystal 16.
Nice.
16 years. Yes, 16 years ago we have installed the first instrument. So we are actually celebrating it, and for all the attendees today that hear me, and if you are interested, it will come soon, a half day or a few hours workshop somewhere soon in April or May. On the crystal 16, and sharing a bit of past experiences and how was the evolution since 16 years ago up to now. It was really nice actually to see an old picture of the crystal 16. Coco, we have a few questions. One of the attendees would like to have also, for example, the reference of the APAP work, but we have a few other ones like What is the minimum solid concentration that can be detected? Oh, this maybe is for for us, that can be detected by the crystal 16. Anything lower than 0.5 milligrams per mil? Yes, it is, and it highly depends. I will answer this, Coco. And it highly depends also on the type of compound. If you I'm thinking of, for example, the crystals that they are transparent type of and very small, indeed, at even a bit less than 0.5, you can still detect them, but lower than that, sometimes it is a problem. In general, yeah, you rarely hear about this type of limitations. Another question, this is for you, Coco, this time. I'm reading it carefully. What is the mole or weight percentage of polymers added in various experiments?
Yeah, so we had 10 micrograms per mil in 20 mil of APAP solution. Yeah, so
Okay.
let's do the ratio, yeah.
Another question, just wondering if you tested different model compound, polymer concentration, and supersaturation level.
Yeah, that's an excellent question. So for APAP, we only tested an initial supersaturation of 1.5. With dipyridamole, I had various initial supersaturations. So if you refer back to my thesis, you will see that there were different starting initial supersaturation levels. However, with dipyridamole, I didn't use any polymers. In this study, we only had one polymer concentration. Dr. Taylor may have done more, so I would either reach out to her or look in the literature, but what I did was only one concentration per polymer.
Okay. Another question, how does the parameter estimation work for nucleation rate? Least square optimization?
Yes, exactly. Yeah. So you could pick different models, different, I don't think I have the equation here, but yeah, it was the least square difference between the predicted versus the simulated.
Okay. And I think that's it. I have seen that I received also a question in the email, but we will answer also after this webinar. The questions we will everything that we have received at info@technovis.com, crystallization.com. We will answer also afterwards. As I mentioned, the recording will be available somewhere tonight, so tomorrow you will receive link to this recording. Please stay tuned. We will have we will celebrate with all of you the 16 years of the Crystal 16 soon. And Coco, once again, thank you very much for joining us, for sharing all your experience and knowledge with us and with our customers today. And hope to see you also in the future and also in person when it will be possible.
Absolutely.
Thank you very much.
Yes, absolutely. It was my pleasure to share my learnings, and I hope that this can benefit one or two people with their research. Feel free to email me directly or Carmen with any questions. I'm happy to help. Thank you,
Yes.
Carmen, and thanks to all of the attendees for their time.
Thank you very much. Thank you all for attending and being today. Thank you for all your questions. Thank you, Coco. Stay all safe and healthy.
Take care. Bye.
Bye.